Thanks, Rosanna, for this really nice intro. And hi, friends. It's fun to be up here after sitting down there for like 20 years. All right, let's see what we can do. It really is a pleasure and a privilege to be part of the OHBM community. And so I really want to thank you all for showing up, filling the seats. So today we're going to have a good time. So we're going to play a game. And I want you to shout out the first thing that comes into your mind when you see this next image. It's going to be a brain image. And really just shout out like what you think the name of this brain image would be. If you were to give this network a name, what would you call it? Are you guys ready for shouting? Okay, all right, ready. Okay, now 1, 2, 3. Please name this network. That was pretty good. So visual network. Okay, ready? We're going to go again. 1, 2, 3. DMN, default mode. Some pretty good consensus. You're warming up. Okay, next one. 1, 2, 3. Whew. Okay, I'll give you one more try. Ready? 1, 2, 3. You're not very good at this. Well, you know, that's okay. We're going to work through this together in the next 38 minutes. Okay. So as you all know, I like to use this video from Daniel Margulies because it's beautiful, and it shows what we do when we're in the scanner resting. There's a lot of spontaneous activity in the brain, and it's not just random. It's correlated in some structured way, and it's kind of remarkable that you can recapitulate things like language, attention, and all these other networks just from this laying in the scanner activity. And thanks to Bharat Biswal, dear friend somewhere in the audience here, we have this method of looking at resting state functional connectivity, and he first published on the correlations between the motor cortices in 1995. And since that time, we've really kind of taken advantage of this approach to think about how the brain is spatiotemporally organized with, of course, the famous default mode hypothesis of brain function over 20-some years ago now. And subsequent network analyses published showing how you can recapitulate a lot of these things, a lot of different networks in the resting state. And then around 2010, we've really kind of pushed the data sharing and big data sets, population neuroscience initiatives like Human Connectome Project, and then we've moved since into exciting territory like looking at the fetal brain, and then of course starting to think about like what are the best practices for looking at data. And now in this current day, we have so many multiple ongoing data sharing initiatives that make all of these data sets available to researchers for discovery science. But I think we're still a little bit behind on a few like basic things, as you all probably just demonstrated to yourselves. Like what is a taxonomy of our functional brain networks? When we talk about them, what exactly are we describing? And I think we have taxonomies in science for naming, describing, and classifying stuff. Because it facilitates organization and communication. It helps us talk to each other and find out what we're doing across labs. So I think the problem that we just illustrated to ourselves about network naming is not just a resting state fMRI problem. I think we kind of inherited it from cognitive neuroscience. And so I'm a cognitive neuroscientist, so I take the blame as much as anyone. But Taylor Bolt in our lab many years ago decided to do some work using maps from the NeuroVault and the Human Connectome Project task fMRI data sets, and he did a bifactor analysis, exploratory bifactor analysis applied to over 100 of these contrasts, and found a general factor that explains about 50% of the variance across many different tasks. And it actually looks, and this is from task fMRI data, but it actually looks a lot like what Fox and Franson independently both published in 2005, this sort of default mode network, task-positive anti-correlation. And of course, there's other subtypes in this bifactor model that explain another 31% of the variance. But I think the point is that even in cognitive neuroscience, we sort of had this problem of not knowing what to name things. And in fact, a lot of times these task-positive networks are called by multiple names in task fMRI literature, like multiple demand, domain general, extrinsic mode, executive control networks. These are all different ways of describing frontoparietal systems. Fortunately, there's some convergence, like you all did pretty well with the default mode network, so that seems to be in a spatial topography that most people kind of recognize. But is it important for us to get these names right? I guess what's in a name? Does it matter? And I think if you look across the literature, there's a lot of cases of descriptions of networks and the anatomical regions that are thought to comprise them. So for example, the cingulompercular network is said to include frontal cortices, insula, dorsal anterior cingulate, and thalamus. But then also a different system named the salience network is also thought to include insula, subcortical structures, dorsal anterior cingulate. And then there's yet another network, the ventral attention network, that seems to include the insula and some of these other regions. And I'm going to focus a little bit on the insula because it's kind of my thing. So I'm going to just use this as an example of why I think some of these network naming problems, are consequential for the field. And so if you look across functional brain network atlases, there's a lot of discrepancy between what different people call the salience network, as one example. And I don't know about you, but that's a problem. I mean, if we're calling something the salience network and across 4 different atlases, this network with the same name has different spatial topographies that have some overlap but not complete overlap, I think that does present a little bit of an issue when we're trying to translate things across labs or use things for clinical translation. And I kind of wrote a book on this, so I really do think a lot about what we call these things and what it means if we're not agreeing on what they are. A while ago, when I was a postdoc working with Vinod Menon's group, we did some work with causal modeling and found this kind of interesting thing where the salience or the dorsal anterior and ACC in particular seems to, you can sort of use signals in it to predict what's going to happen later in the lateral frontoparietal, what some people call central executive networks, and the medial frontoparietal or well-known default networks. And I think more broadly, the insular cortex is kind of just a very interesting region for a lot of us. In particular, when you look at the cognitive neuroscience literature, it seems to be active across things like emotion, empathy, and pain. But at the same time, if you look at other studies, it's involved in subjective awareness, interoception, and somatosensory processes. And then, of course, high-level cognitive processes like conflict, switching, attention, and inhibition. So it's really one of those areas that we all kind of love to look at. There's a nice paper from Thomas a long time ago now using hierarchical Bayesian modeling applied to the brain map database and looking at sort of cognitive components that make up different task activation maps. And he reported back then that the anterior insula seems to be one of those really functionally flexible brain regions. Like it's involved in a lot of different cognitive components across a lot of different tasks. And we did this work a while ago with Luis Pessoa and Michael Anderson, looking at the functional diversity of the insular cortex. And if you look at the dorsal anterior insula subdivision in particular, if you calculate this measure of diversity, which is basically how many different types of tasks that this region is engaged by, the dorsal anterior insula has kind of the highest level of task diversity compared to the other regions. So it's really involved in a lot of stuff. And so if you use what a lot of us like to do is look at brain dynamics, and we like to use Vince Calhoun's nice toolbox GIFT to use sliding windows to look at how connectivity can change from moment to moment and compute things like frequency of occurrence of brain states, dwell time, and state transitions. And of course, other methods like coactivation pattern analysis that can also be used to look at dynamics across time. A while ago, we also used the sliding window approach to kind of look at the insular cortical dynamics to see if that, functional dynamics of the region could help explain some of its functional flexibility. And so this is just from resting state fMRI data, and no point in even trying to read the slide. But the take-home message here is that there's some functional connectivity states in which different insular subdivisions are talking to different areas of the brain, and there's others in which they're more similarly functionally connected to the rest of the brain. And I think for example, in state 3, the insular subdivisions are kind of talking similarly to other brain regions, but in state 5, they diverge in their functional connectivity, which happens less frequently. But what's most interesting, I think, is that the dorsal anterior subdivision is the one that's the most functionally dynamic in terms of its connectivity partners across time. So it's, again, like we showed in the task stuff, it's the most diverse in terms of what it does and who it talks to. And we had a great summer student, Will Snyder, in the lab a few years ago, looked at these insular cortex dynamics across the lifespan in the NKI Rockland data set and found some, again, like interesting changes across the lifespan in terms of the insula and ACC and how it connects to the rest of the brain and how that is different depending on if you're in the younger part of life, in midlife versus in later life. And finally, for thinking about insular cortex dynamics, I think it's also, of course, relevant for those of us interested in clinical translation and neurodevelopmental disorders. My lab focuses a bit on autism, and so we took the co-activation pattern analysis approach, and this was done by Emily Marshall, who was a med student now, was an undergrad at the time in our lab, and she just took the data from the data we'd collected in 6 to 12-year-olds with autism, combined with some of the autism brain imaging data exchange, and found that there was a co-activation pattern like involving salience, executive, and default mode networks that showed a reduced frequency of occurrence in kids with autism compared with typically developing kids. And so what I actually, I'm not sort of focusing on the findings here, but more on just like the fact that the insular cortex is, an interesting area of the brain to study across all of these different populations and across the lifespan. And I think if you're interested in biomarkers for precision medicine, it does seem to be one of those brain regions that's the hub and very important and good to understand. Now, I didn't make this figure, but it just so happens to have the insular cortex as one of the brain markers that would be interesting to understand from an RDoC perspective when we're talking about transdiagnostic, markers that could talk about psychopathology, across different diagnoses. I think thinking about insular cortex structure, function, and dynamics seems to be a good way to go. But it's not just me who is kind of really into the insula. A lot of you are working in this area as well. And I remember this paper that was like a VBM analysis from Amit Ekman's group a while ago. And it just showed that across, I don't know how many different psychiatric conditions, there was consistently, no matter what the condition was, there was atypical, morphometry in the anterior insula and ACC, as I hope you can see there. And I love this paper from Chuck Lynch that just came out in 2024 with Connor Liston, where they talk about how the salience network, here the insula and ACC again, right, talk about how it's expanded in individuals with depression. And fun fact, Chuck was a research assistant in the lab when I was a postdoc at Stanford, so I just take credit for everything he does from the rest of his career. But in any case, it's really fun to see, like, there's so many different areas in which the salience and insula, the whole system, is implicated across a range of different psychiatric conditions. And this is some nice work from Mike Fox's group. They do a lot of this lesion network mapping. That's very cool. But again, this is showing how there's a circuit that when you disrupt it with TMS, it's a common circuit for different types of addiction. And hey, guess what? It's like, again, the insula and ACC. So I think sometimes when you look at specific findings, you might not see all these like overlapping patterns, but this is just kind of a sprinkling of an overview of a few studies just to give you the sense that I think it is important to talk about these networks in a standardized way, to think about what we mean when we say salience network or default mode network, what brain areas we're including, and how a sort of misuse of some of these labels could lead to difficulty in replicating work or difficulty when we try to go the clinical translation route. Like if we're really trying to target the salience network with TMS or with some other intervention, we probably should agree on what it is we're talking about. So how are we doing? Okay, so how is a functional brain network defined? I think that's sort of the big question, or one of them. And how can standardization of network nomenclature impact clinical translation? I think it might, or it probably will. So what happened? From now on, I'm just going to tell you like anecdotes, so I hope that's okay. So in about 2018, found myself at the Alpine Brain Imaging meeting with my good friend Nathan Sprang, and I took my first and my last skiing lesson, and so that was it for me. But anyway, it was a fun conference. They asked us to write something at the end of it. So we cooked up this idea that we would start to try to build a universal taxonomy of functional brain networks for all the reasons I just kind of described. And then we thought we really need to add someone more legitimate to the team. So we brought in Thomas Yeo. He thankfully agreed. We thought, yeah, we really need an engineer. And he's got those nice networks. And so we brought him in, and we said, okay, let's just look at what is going on in the literature across these different atlases. And there's a lot of them. This is just a small survey, but most atlases that are out there have something in the back of the brain that is often called a visual network. And many of you agreed, because when we showed you a visual network, you're like all over it, right? So a lot of the parcellations have visual networks, and they're easy enough to label. You see them in Evan Gordon's parcellation. You even see them in one of these papers, ICA paper looking at task and rest from Steve Smith. So instead of calling it a visual network, we thought, why don't we just call it an occipital network? Just go with the anatomy. Yeah, it does visual stuff, but let's just stick to the anatomy for now. And if you go through and look at all these other systems that are often delineated, why don't we just stick to the anatomy? So if it's the motor system, motor network, let's just go with pericentral network. You can see it across these different parcellations. So you see where I'm going. We figured let's just try to stick to the anatomy. We'll make that work. Dorsal frontoparietal network instead of dorsal attention network, because yeah, it does attention, but maybe it does other stuff, who knows? So we tried to kind of get this kind of anatomical scheme going. And of course, we went through and found, there's always going to be a lateral frontoparietal network. People will call it all kinds of things. So maybe we should just stick to the anatomical terms. And again, this one's kind of a mess, but the salience, singulo-opercular, ventral tension, whatever, let's just call it mid-singulo-insular as far as anatomy goes, and we'll work the rest out later. This one, everyone's happy with the default mode network. I guess no one really wants to relabel it. But if we were going to go with this relabeling scheme, we would probably call it the medial frontoparietal network. So I thought, not bad for a preliminary taxonomy of functional brain networks. In terms of standardization, if we could get everyone to agree to call them by anatomical names, maybe we would have a little bit more replicability and consistency. So for a few years, this was 2019, for a few years I just every paper we published, I used this nomenclature, and literally no one else did it. So it was not even Nathan. So, this is yeah, okay. We tried, or at least I tried, but it wasn't going to work for a lot of reasons, and I think it's pretty clear what some of those reasons are. What about dynamics? We saw a beautiful talk from Joanna Cabral yesterday. We all know that things change, and whatever networks we're talking about are probably just a static approximation of a much more complicated dynamic system, and we're just not really capturing that very well in our static nomenclatures that assume strict set number of networks when they're really moving around a lot. What about subcortical brain regions? We kind of just ignored the subcortex, as people tend to do. What about all this inter-individual variability? We've seen a lot of this deep phenotyping suggesting that some of these network boundaries are not super strict and can move around a bit. And we just saw the stuff about salience network expansion and depression. So, things change between individuals. And then, of course, there's this question of scale. Clustering is just whatever you tell it. So you can have a 7-network solution. You can have a 17-network solution. I believe, Thomas, in your paper, both of them were equally stable. Even a 9-network solution was probably equally stable, but for some reason the 7 stuck. So, clustering is great, but it's not going to give you the ground truth, right? So you can always fractionate, and you can get into subsystems, and there's hierarchies. Where do we stop? So this is kind of a mess. So we didn't stop there. we kept going. But just to like go back in time a little bit, if you remember before the days of MRI, we still had a lot of nice thinking about network neuroscience that, kind of took into account a lot of these issues, like the many-to-many problem, that cognition is served by interconnected networks, that complex behavior is mapped to the level of multifocal neural systems rather than specific anatomical sites. And so we knew this even from, back in the day, like neuropsychology. We knew this from lesions, that like many brain regions contribute to many functions. There's not a one-to-one mapping. We knew for a while from nice work by Randy McIntosh and others that it's important to consider the context of what's going on in the rest of the brain when you're trying to understand the function of a particular brain or particular network. The functional, this idea of neural context, right? The functional relevance of a brain area depends on the status of the other connected areas, and we can't sort of ignore that context. And of course, we've really begun to take more seriously the idea of dynamics, and in terms of, this is a nice quote from one of Louis Passeau's papers. I really like his concept of dynamic affiliation. Some brain regions like the amygdala might dynamically affiliate with a network for some amount of time and then affiliate with another one for some other amount of time, et cetera. So a network needs to be understood of the interactions between multiple brain regions as they unfold temporally. And here's Taylor again with another great paper. This is when he was looking at the HCP resting state data set and found this kind of 3 spatiotemporal patterns that constitute the dominant spatial structure in spontaneous bold fluctuations. And basically, it was kind of interesting to see that previous observations like Daniel Margulies' famous gradients and anti-correlations between default mode network and test-positive networks and various time lag propagations can be explained or sort of as manifestations of these 3 underlying spatiotemporal patterns that can be captured with complex PCA. Ask him if you want more details about this. I don't know how he did it. But the point is there's so much of this complexity in the brain that we sort of tried to ignore when we put these labels of networks on stuff and try to kind of move forward with that. So there was some very wise reviewer who, after reviewing our paper on that structural, or on the anatomical taxonomy, they said, why don't you form a work group and you can get together a bunch of people who are experts in atlases and networks and philosophy and clustering and try to solve this problem once and for all. And we said, great idea, let's form a work group. So we got the best and the brightest together. We got Anastasia Hindiki to come up with an acronym, and I think this was amazing. So she came up with the Workgroup for Harmonized Taxonomy of NETworks, right? WHATNET. And then we got all you fine folks at OHBM to give us a stamp of approval as a best practices committee. So in the grand tradition of COBIDAS and the best practices committee on data analysis and sharing, and then the MEG wonderful committee that was chaired by Agna Pius, we thought we're gonna do what they did and have a best practices committee on naming of networks, and then we're gonna really solve this. So what did we do? We met over Zoom starting in 2020 for about 3 years, once a month. We spent probably the first year talking about what is a network, or is there a network, or does a network exist? There was a lot of that. And then we kind of decided there were networks. Finally got that philosopher to calm down. And then we love Felipe. But so basically, this was the group. I mean, I think most of you are here in the group, so you know who you are. We wrote this really long paper talking about what are some of the problems and how can we resolve them in terms of getting towards this more harmonized network taxonomy. What are the issues? We talked about the intersubject variability. We talked about, should we only use fMRI as the standard? Should we look at other imaging modalities? All those things. It was good times. And then we did this survey, kind of like the one you all did at the beginning of this talk. So we did send it out and I asked people, like, what is this network? What's that network? And just as you all were terrible at it, so was the internet. And but there was some good things, like there was the default network, the visual network, and the motor network. People pretty much agreed. There was like a lot of agreement when we showed those images and we said, what is this? Most people were like okay with visual, motor, and default. They were not okay with the frontoparietal, salience, singulopercular, all of these other ones. They were not agreeing. So, you know, that was the survey. So we did our minimal reporting guidelines in that 2023 paper about, you know, when you're reporting results, what are some minimal things you should try to adhere to, kind of like COBITUS, but like not so much because there's only 6 guidelines. But the idea is like, you know, when you're using task fMRI and have GLM results, like it's not necessarily a network, but I mean, you can call it one, but you know, there's that. And we tried to, you know, suggest that maybe you should avoid labeling patterns of brain activity or connectivity with just like some idiosyncratic term, like maybe just, you know, don't make up a brand new thing. And then, you know, we kind of suggested that people should really take seriously the idea of indexing novel findings using reference atlases to show that, you know, the overlap with these certain different parcellation schemes, just to make it easier. I'll talk more about that in a second. We also thought it'd be important to report the sample variation from the population used to generate the reference parcellation scheme. So we all love the atlases that are out there, but they're mostly like 20 to 30-year-old adults, you know, very, you know, not super diverse samples, and that's fine, but I think people kind of forget that sometimes in the, you know, then using those same atlases to try to report on other populations, younger or older and all of that. And then I threw this in there, consider supplementing the labels with additional anatomic network labels if you want. Nobody's actually doing that, but, and then we suggested that you follow whatever Tom Nichols says to do in COBITUS, because that's probably a good idea. So we thought, great, OHBM best practices gave us a stamp of approval, and we're done. And yeah, we were not done. Okay, but I still have a few minutes. So what did we do? Well, I went back thinking more about this problem of the grad student and the brain map, right? Like, what do you do when you find something and you just like don't know what to call it, and you bring it to your PI and they, you know, if your PI is someone who likes the salience network, they'll be like, you should call it the salience network, and that's kind of what goes into your paper. And if your PI is Thomas, you'll call it the salience ventral attention network and move on with your life. And, you know, if you're in another lab, like, things could change, you know, people change their minds. So, you know, it's kind of hard to know, like, what to call something, you know, when you're starting out in the field and you're just trying to write a paper, you know, you're really just, you just want someone to like tell you what to do. And we're not here to tell you what to do, but we are here to introduce, wait, where is it? Oh, okay. The Network Correspondence Toolbox. This is the best thing. I'll tell you why. Because, so Ruby Kong in Thomas's lab spent a whole lot of time doing this, took all the atlases that are there, started with 16, then like 22, and you can, you know, we're still adding more atlases to it. But the idea is you can just add all these atlases into this, load them into this toolbox, load whatever map you have, and you can compare basically your data file with all these available atlases and just like compute a simple dice coefficient to show you like the amount of overlap, and then you can report out there's like this much overlap with this much network in this atlas, but also that much overlap with that network and that atlas. And like she spent a lot of time making this nice interface, and it's a Python toolbox, you can download it, it's all there. So, oh yeah, and if you want to play around with it, like you can take like, for example, Evans and Thomas's parcellations and compare them to each other. And I love this because it helps, you know, disambiguate some of the stuff I was talking about earlier in the talk. Like I think like Evans' singulopercular overlaps a lot with Thomas's salience ventral tension A and B, just in terms of spatial topography. And that's good to know, right? So I think this is fun. I encourage, you know, looking at this. But you can take your own input data, right? So whatever that yellow map is. Let's pretend that's yours. And you can specify which atlases you want to compare it to. It could be all of them. It could be 2 of them. And it just computes this dice overlap and gives you these nice network clocks and network radars, which tell you, like, for each atlas, how much overlap is there between your thing and, the other thing. And there's, like, p-values and various nice stats in there as well. So this is like an easy example, right? So if you look at the Human Connectome project, this is a 2 back-working memory task, like easy example. You stick that in the toolbox, and it tells you, for example, like, a lot of these atlases call that the dorsal attention or frontoparietal, and it just gives you an easier way of coming up with how you want to talk about this in your paper. You could even just use this figure in your paper, right? It's just a little bit, straightforward. And then sometimes you get these things like this is a UK Biobank ICA map that's like not so easy to parse. And so instead of guessing, you can just stick it in the toolbox and say, well, here's what the toolbox says. And it's like a decoder. And that's what Transmitter called it. I didn't tell them to, but that's what they called it. So it's a decoder tool, and I think it takes, a little bit of the guesswork out of what you want to call things. So that's where we're at with that right now. And I really want to thank the Open Science folks for this nice write-up, because I don't have to summarize because it's all summarized here. So, when we have all this variability in brain network definitions, it does make it hard to translate findings across fields. It makes it really hard for, neurologists or psychiatrists who are trying to parse the literature and try to decide if whatever they're reading applies to their clinical population. So this kind of quantitative evaluation of brain network correspondence that's facilitated by the toolbox, I think, will help, align results, for future interpretability. There's also statistical validation for comparisons, cross-atlas comparisons, which will, help us improve our accuracy in reporting, and a user-friendly design and, hopefully easily accessible to help us get towards this sort of advanced standardization in neuroscience. So, as the field of network neuroscience matures, it'll become increasingly important and imperative for us to arrive at some kinds of consensus about nomenclature, but it doesn't, we'll see about that. But at least the reporting of results we can facilitate with the toolbox, which helps us, I think, in the future translate findings across labs while we work towards, a little bit more of this universal taxonomy. This is from Glasgow, where we had a nice WHATNET reunion, brought in all our trainees. We all got COVID, but, it was worth it. What's next? You can, there's always new atlases. They just keep coming, right? So this is, I think, a list of, I don't remember when this paper is published, but there's a lot of them out there, and so we want to make the toolbox, or the toolbox is such that you can upload new atlases, you can upload your own or others, so you can kind of keep adding to the toolbox, and it'll, be as useful as you make it, essentially. Ruby's really great about responding to people when they've come with questions or suggestions. So it's a work in progress. I know a lot of folks are working on infant atlases or different age groups. This is, I know, Vince Calhoun and others are working on these ICA-based atlases to try to get at some more consensus from the, we didn't talk about ICA much today, but there's other ways of creating atlases. And Gaël Doucet's group is working, or has published, like an adolescent reference atlas using ICA as well. I think there's a lot to be said here that I'm not going to say, but about the fact that oftentimes we use standard atlases in populations where they may not be the best reference atlas, like in very young kids or in aging brains or other populations. So I think like the more we develop new atlases and kind of incorporate them into some standard framework where we can compare with other existing atlases, the better off we'll be. And I think we really haven't addressed this issue of dynamics and contextual issues in our network framework. This was a really nice paper from Rasko Sirik a while ago when he was, I think, an undergrad working with Ajay Satputty, and he did a nice study looking at how basically static localized networks are superordinate approximations of these underlying dynamic states. And so and each of these kind of localized dynamic connectivity states is associated with global changes in the whole brain functional connectome. And so I won't sort of go through this today, but it was really interesting paper, and it just kind of shows how much we're not doing when we kind of put a static label on a functional network, how much we're kind of ignoring. We all sort of know that, but we have to do it anyway for communication. So towards the universal taxonomy of functional brain networks, we're towards it. We're still moving towards it. I think 6 years later. Are we there yet? No. But we definitely are making progress in terms of suggesting ways to communicate findings that will help us talk across labs and do more reproducible work. And in terms of, are we there yet? This is another fun study from Taylor that I'll just leave you with. It turns out if you look across the corpus of all the literature, hundreds of thousands of studies, and you look at the phrases that come after the term, come after the phrase "the brain is," the most popular terms that come after that phrase are an energy-demanding organ, a complex dynamic system, a complex network, a heterogeneous organ, a control system. So, I guess it's no wonder we haven't solved it yet. It's complicated. So that's kind of where I want to leave you, and hopefully we'll have some time for discussion. But I've been here almost 20 years. I love this place. This is great. It's just wonderful to be up here with all of you. This is my lab. Thanks to them. Talk to them if you see them here. And also, great news, ABCD 6.0 data release just dropped. So that's happening right now. Get on your data use agreement. And if you have also given up skiing but are really into surfing, come to NUSA in February of this next year, and that's where we'll be. Thanks, everyone, and funding, et cetera. And come to some of these posters if you have a chance. All right, that's it.
Thank you so much, Dr. Uddin. What a fantastic and comprehensive talk. So we got a lot of questions. We might be able to go through a few. I'm just going down the list in terms of popularity, and also, there's some common themes. First up, does the functional diversity of the insula reflect in connectomic, receptor, and/or cytoarchitectonic diversity?
I think so. We definitely find unique structural connections in the dorsal anterior insula to the frontal and other subcortical regions. I'm not very well versed on the cytoarchitectonic stuff, but I wouldn't be surprised if that were the case.
Okay. So as a follow-up to that, so there's a couple that go together. One is could the multiple network definitions be a consequence of trying to spatially constrain a network that might be dynamic? Or relatedly, at what point can you define something as a network?
Yes.
So what are the implications for application? Because for many years, there's been a push for networks to be a biomarker, something that's robust and definable, interpretable, all that?
Yeah, I think we always approximate the networks as static, even though we kind of know they're not. But I think the implications for clinical translation are really huge, right? Because it's possible it's the dynamics of the network themselves that are differentiating some of these disorders, but when we do the static snapshot, it looks like it's all the same network. So I do think we have to move forward a little bit in our thinking about what is a network, across time, milliseconds and also across lifespan. But I think that's the next challenge.
Yeah, good answer. So another question is that here at the OHBM, we don't all do MRI. So do you think a similar set of definitions could be developed for the other modalities, such as MEG, EEG, FNIRS?
Yeah, definitely. we had a couple EEG experts on our WHATNET group who said exactly that. And I think we tried, I mean, basically we haven't gotten far enough along the consensus building, even in fMRI, much less other modalities. But I think we definitely need to kind of work harder to integrate a lot of these different, tools into our thinking. But yeah, we're a little bit fMRI dominated at this point, unfortunately.
Okay, thanks. That's all we have time for. So if you didn't get your question answered, please come up to the stage after or submit it to the app. Let's thank Dr. Uddin again. Thank you, Lucina.
Thanks.